Welcome to Part 3 of the CMT Research Foundation's CMT 101 series. In this webinar, I'll be discussing therapy development for CMT. The pace of therapy development for CMT is increasing. The good news is that we have 21 therapies in various stages of development in industry for CMT. How do these therapies go from the laboratory to clinical trials and eventually your medicine cabinet? Every drug in this country has to travel the same road to final approval by the Food and Drug Administration or the FDA. On average, this is a 12-year process. Let's take a really simplified look at what the process would look like in CMT. First, scientists need to gain a better understanding of the underlying disease mechanism through identifying the gene that is causing a specific type of CMT, or by identifying the pathways that are altered in nerves or Schwann cells and lead to pathology. Then a target in the cell needs to be identified, which could be potentially modifiable by a drug. Targets are generally genes or proteins. Next, scientists screen for drug candidates in models of CMT. These models can be cultured cells, typically adult stem cells which are differentiated into nerve cells, or animal models, typically mice or rats. A good candidate for future tests in humans will be able to modify the nerve pathology in animals and not be toxic. Once this data is in hand by a company, they will submit the data to the FDA, and FDA evaluates whether the therapy can move ahead to be tested in people. In a phase 1 clinical trial, the candidate is tested at several doses, typically in a small group of people without CMT, but not always, to determine whether it's safe for humans. If the safety profile looks good, then it's on to phase 2 clinical trials. Here, the drug candidate is tested in a small group of people with CMT to determine if the drug is effective in causing beneficial changes that can be measured by researchers. For example, do nerves function better? Has muscle strength changed? Can somebody walk faster or farther? Further tests are then done in a larger group of patients in phase 3, again measuring if the drug is causing beneficial changes. If the data shows a beneficial change in the disease, then the company submits a new drug application to the FDA for their review. FDA experts evaluate the data and determine whether the drug is approved. Therapies for any disease fall into one of two categories. They either treat the symptoms or actually address the underlying cause of the disease. The latter is called disease modifying. Disease-modifying treatments can slow down, halt, or even reverse or cure a disease. Most of the drugs in development for CMT have the potential to be disease-modifying. At the CMT Research Foundation, we are very much interested in funding science that leads to disease-modifying treatments. 3 general classes of therapies are being developed currently for treating CMT in industry. Small molecules are chemicals that are synthesized in the laboratory and usually given systemically. Depending on the specific drug, these treat either symptoms or are disease-modifying. For example, small molecules could slow down or prevent decay of axons or myelin, or provide more energy to nerve cells to make them function better. For cell therapy, cells are isolated from different human tissues, purified, and then injected locally near specific nerves to enhance myelination and make nerves conduct electrical impulses better. This approach would be considered disease-modifying in a limited number of regions in the body, depending on which nerves were targeted. Lastly, gene therapies target the underlying cause of disease by adding, removing, or modulating genetic information in nerve cells or Schwann cells. Gene therapies are delivered via modified viruses, usually AAV, or synthetic nucleic acids called antisense oligonucleotides, or ASO for short. These therapies can be administered locally or systemically. One current challenge with these various gene therapies is that the peripheral nerves are not efficient at taking up these viruses or ASOs. One of the research priorities for the CMT Research Foundation is to fund projects that solve this delivery challenge. Gene therapies can be broken down into 5 types of approaches listed here on this slide. In the first, gene replacement, one would add a normal copy to replace the mutant gene that is not working in an individual. With gene knockdown, one aims to inactivate the mutant gene. With gene editing, one actually corrects the mutation in a person's DNA. With gene knockdown and replacement, one first inactivates the mutant gene and then adds back a normal copy to the DNA. And finally, in gene augmentation, one bypasses the mutant gene entirely. It's important to match the proper gene therapy approach with a specific CMT subtype. Gene mutations in CMT alter the encoded protein in different ways. One can have an inactive or missing protein, a toxic protein, or too much protein. Each of these scenarios requires a different gene therapy approach. For example, in the case of an inactive or missing protein, gene replacement would be an appropriate therapy to use. In contrast, in the scenario with too much normal protein, one would need to employ gene knockdown or gene inactivation to fix the problem. This is why it's important to identify the specific mutation in a patient and understand the effect of the mutation on the protein. That's the type of work that's done in the discovery stage of drug development. The primary cell type that's affected in axonal types of CMT, so CMT2, is different than the cell that's affected in demyelinating forms of CMT, CMT1, 4, or X types. So it makes sense that different therapeutic strategies would be employed for axonal versus demyelinating forms of CMT. The potential exists for drugs that are developed to treat one type of axonal CMT to be used for treating other types of axonal CMT. For example, drugs that block axon degeneration. Or a drug that's developed for one type of demyelinating CMT could be used in another form of demyelinating CMT. This could be a drug that blocks myelin degeneration or stimulates myelin repair. Methods that enhance delivery of genetic therapies to the peripheral nervous system also could be used in multiple forms of CMT. Let's look at the different therapies in development for the demyelinating CMTs, of which there are 13 candidates. There are some general take-home messages in this pipeline. The majority of the therapies that are under development are in development for the 1A subtype due to both the underlying biology as well as the market size. CMT1A is the most common form of CMT. Only one of the therapies is in development for a CMT4 subtype and none for CMTX types. Most of the therapies are either small molecules in green or gene therapy in blue. The therapies are found throughout the development process from discovery to phase 3 clinical trials. The longer the bar, the farther along in the process is that particular therapy. Let's move on to discussing therapies that are in testing for the axonal types of CMT, CMT2. There are fewer candidates here, only 7, because it's a smaller market size. Most are for 2A, which is the most common type of CMT2. These drugs are earlier in the development process, and most of these therapies are small molecules in green, and only one is a gene therapy in blue. The CMT Research Foundation stays abreast of these pipelines, and we're in regular contact with many of the companies listed here. We support their work and look for ways to complement these pipelines. Additionally, we're working to solve some of the obstacles that some of these drugs face to get to approval. There are 3 main take-home points for this webinar. The first is that various therapies are in development for CMT. Second, therapies can either treat the symptoms or the underlying cause of CMT. The latter is called disease-modifying. Disease-modifying treatments can slow down, halt, or even reverse or cure CMT. And finally, therapies have to be tailored to specific types of CMT. If you would like more information on any of the topics covered in this CMT 101 series, please contact us through the links on the CMT Research Foundation's website.